Hello everyone, my name is Mara Cloutier. I'm a PhD student at Penn State University, and I'm going to be walking you through the PICRUSt2 workflow and getting you ready for analysis either in R or in QIIME 2. If you're here for dog, you can click on the Here button on the right hand corner to get the documents if you want to run through the tutorial. If you want to see more information about PICRUSt2, you can go to the GitHub link here, or you can see the paper that is in preprint on this link here. So before we start PICRUSt2 from R. So in R, if you've got biome format loaded, then you can just take your, if you're using a phyloseq object, then you just take your OTU table from your phyloseq object, export it as a matrix, and then transform it into a biome table, and then export it as a biome table. If you're, again, if you used data 2, then what you should have ended up with is a sequencing table that has all of the sequences in it, and then you can just output that sequencing table using uniques to FASTA. And you want to output it as whatever name file.fna that you want, and you're going to ID them based on the column name. So what that's going to do is it's going to use the ASVs as the names of each of the sequences, which is exactly what we want. And so there's one problem with the resulting output file that I wasn't able to figure out how to manipulate in R, so I'm going to walk you through how to do it just using Excel, and it's very simple and easy. So in your first line of each entry, there's ASV number whatever. After that, there's the semicolon size equals some number semicolon. Well, this is a problem because your biome file doesn't have these semicolon size equals semicolon after each of the ASVs, so you're going to run into issues where you can't run the commands in PICRUSt2. So you need to get rid of the semicolon to semicolon section. The easiest way to do that is to just copy and paste the FNA file into Excel and then remove that section and then copy paste back into the FNA file, and everything should run smoothly. If you haven't already, then you'll need to install PICRUSt2. If you're running a Mac and you already have Conda installed, then it's incredibly easy, where you just run conda create PICRUSt2 and everything should work smoothly. And then after you've installed it, then just conda activate PICRUSt2 and you should be good to go. So you'll need to check both the sequencing file and the biome file to make sure that they are in fact in the right format, and so you can do this quickly by saying less repseq1.fna or whatever name your sequencing file is, and it should look something like this on the left, and then you can run biome head-i and your input biome file, and you should see formatted file, something like this on the right, where you've got ASVs going down the rows and then your samples across the columns. And so that will be the end of this short tutorial, and when we come back, we will actually start to run the PICRUSt2 commands. Okay, so once we've made sure that the input files are in the right format, then we can start running the PICRUSt2. So the first thing that we need to do is is to build a phylogenetic tree. And so we do this by taking our ASV sequences and aligning them against a reference or database of reference 16S sequences using HMMER, hidden Markov modeling. It's a very common modeling tool to align sequences. After alignment, we're going to take our queried sequences and place them into a reference phylogeny This phylogeny that we're going to be referring to is based on the 16S sequences, and it's in a Newick format tree. And in this tree and in the reference database includes 20,000 16S sequences from the IMG database. So it's a lot of sequences for 16S. It's important to note, though, that most of these 16S sequences are going to be from human gut bacteria. And really, PICRUSt was initially created to predict functions of human gut microbiota. But to make this phylogenetic tree, we're going to use placeseq.py with our sequencing file, and we're going to output a tree. If you want to, you can pass the parameter p, where you change the number of processors. Once we have that tree, then we can use it and predict the enzyme commission, or EC number. This can also be used to predict KEGG orthologs and many other parameters as well that we will talk about later. And these EC numbers can be used in the Metaseq pathways. So if you're not familiar with Metaseq pathways, it is a database online that includes many different metabolic pathways. The one that I have showing on the bottom right is for dissimilatory nitrate reduction, and you can see there's many arrows going from one enzyme or a protein to another, and each of these arrows represents an EC number. So these are really enzyme numbers that fall into some sort of metabolic pathway that can be found on this website. So to do this, you pass the command hsp.py, where we're going to be calling ESV numbers using our tree, and the output file is going to be this EC predicted tsv.gz file. And then again, you can pass processors to be whatever number you are comfortable working with. This command does take a while to run, so just be prepared to wait it most likely a couple hours depending on how many samples you have. So you can unzip that .gz file and open it up in Excel, and it will look something like this file here, where we've got ASVs going down the rows, and then we've got EC numbers going across columns. And so how to interpret this file is that for ASV1, it's predicted that there will be 2 EC 1.1.1.1 per genome for ASV1. And it will have 0 EC 1.1.1.10 enzymes per genome, and so forth. So looking at this file, the greatest number is going to be, I think, 7 here for ASV 10,001 and EC 1.1.1.100. And I'll show you how to better interpret this in a few minutes. So many of us already know this, but microbes have variable numbers of 16S rRNA gene copy numbers, and they're hypothesized to be species, possibly even strain dependent. For example, Escherichia coli has 7 copies of the 16S rRNA gene per genome, whereas Bacillus subtilis has 23. So it varies quite a lot between bacteria, and really is something that needs to be accounted for when we do these metabolic function predictions. So one thing that we're going to look for is also, so what we're going to do is we're going to predict the number of 16S rRNA genes for each ASV. So we can account for these differences in gene copy numbers, so we can get an idea of how many genomes there are rather than just how many 16S rRNA genes there are. Another thing that we need to account for is how close the ASV is to the actual reference 16S sequence. And so we're going to calculate this nearest sequence taxon index value, where the closer the distances are, the more accurate the predictions are, right? So if we have an ASV that's aligned very closely with the reference 16S sequence, then we can hypothesize that the functional predictions are going to be very similar. But if we have an ASV that did not place well with a reference 16S sequence, then those functional predictions may be less accurate. So we're going to do this by passing the command hsp.py, and the input, what we want to predict is 16S, and we're going to take our output tree as input, and the output is actually going to be this marker predicted nsti.tsv.gz file. And again, pass this processor's number or however many. And then we also want to pass the command calculate NSTI to give us an idea of how well the ASV is aligned to the references. The resulting output file is going to look something like this, where we've got ASVs going down the rows, and the predicted number of 16S rRNA genes per genome. So for ASV1, it's predicted that there will be 3 16S rRNA genes per genome. For many of these, you can see that it's 1, and then down at the bottom you see that there's 5 16S gene copy numbers predicted for ASV 10,012. We also see these NSTI values here, where generally they're low, but then we have a few values that are higher, like for ASV 10,003, where we have an NSTI value of 3.3. As I mentioned earlier, there are other options that you can use besides just EC. You can use KEGG orthology, PFAM, TIGRFAM, or PHENO, as well as COG. And those can be, if you want more information about that, you can go to the GitHub site on this slide. Okay, so the next step is to calculate the expected number of ASVs and expected number of ECs for each sample. And so how this works is the command takes the ASV table, so basically our biome file, and it divides the number of occurrences of each ASV in that table by the number of predicted 16S rRNA genes for that ASV. And it multiplies the resulting value by the predicted gene family copy numbers per ASV. And I have some calculations so that I can better explain what's going on in this command. And so for this specific command, it's going to be the metagenome pipeline.py, where the input file is a biome file, and we're going to output a ESC predicted.tsv.gz file and an EC metagenome file. We're going to stratify the output so that it's easier for us to look at. So these calculations are only a little bit complicated. So let's say in sample 1, we have 36 sequences assigned, 36 16S rRNA genes assigned to ASV3. So basically, when we're looking at our OTU table and we're looking at sample 1, there's 36 sequences that are associated with ASV3. Okay? And through PICRUSt, we've predicted that there should be 12 16S rRNA genes per genome of ASV3. So if we're looking at these, we'll say cells here, the green cells, they're all going to be ASV3, okay? Because I have 3 of them because it's predicted that there's going to be 12 16S RNA genes per genome. So that means for one ASV3 cell, that genome is going to have 12 16S RNA genes. And if we've got 36 total rRNA genes in sample 1, then that means that we should have 3 cells of ASV3, okay, where the 16S genes are the gold stars. And we've also predicted that there should be 5 EC3.3.3 enzymes per genome. Okay. So if we have 3 cells and 3 genomes, then we should see, or we can predict that there will be 15 of these EC3.3.3 in sample 1. So then the total number of expected EC3.3.3 in sample 1 is going to be the cumulative sum of the last column of this table, right, where we have 15, 21.7, and 28.6 to get 65.3. So the output files from this metagenome pipeline, there's 4 output files. So there's a seq-tab norm CSV file that has the ASV abundance table normalized by the predicted 16S gene copy number per ASV. So what this is basically this is a normalized ASV table that takes into account number of 16S gene copies per genome. Right? And then we have a weighted NSTI value table where it gives you the mean NSTI value per sample. The PICRUSt developers have suggested that NSTI values between 0.06 and 0.15 are ideal, but it is going to be highly dependent on your samples and where they came from and just how well your sequence is aligned with the reference sequences. So the other 2 output files are a predicted metagenome unstratified TSV table where is the overall EC abundances per sample. So this is essentially like an ASV OTU table where instead of ASVs or OTUs, there's EC numbers. And then they have abundances per sample, and so you can just take this table and start working with it in R or with QIIME 2. Then there's this other file, which is the predicted metagenome contribution TSV file. And this is very similar to the table that I was showing you earlier, where we have the calculations of ECs per genome, where if you look at the second line here, we've got 195S1, so that's our sample, and then we're looking at this function EC1.1.1 for ASV 2012. And then there's this 25.0 for taxon abundance. Now this represents the normalized ASV counts, right? So this is not the raw sequence counts. Now, this has already been normalized. So this is saying that there's expected to be, we're going to say 25 cells of ASV 2012 in sample 195S1. And then there's this taxon relative abundance that is the relative abundance based on 100%. So for ASV 2012, that's 0.2511. And then we have this other column where it's genome function count, and it's 3. So that means that it's predicted that there are 3 EC1.1.1 per ASV 2012 genome. Okay. And from that, we can calculate the abundance of EC 1.1.1 of ASV 2012 in sample 195S1 by taking 25, which is the taxon abundance, and multiplying that by the genome functional count 3 to give us 75. So what that's saying is that for sample 195S1, there's expected to be 75 EC 1.1.1 from ASV 2012. And then we can go one step further and calculate the relative abundance of that function based out of 100, which would be 0.75. So now that we've calculated predicted EC numbers per sample, and also EC numbers predicted per genome for each ASV, we can move on to predict these metabolic pathways or metacyc reactions. Okay. So what this does is it groups the EC numbers into metacyc reactions, and it's inferring what pathways are present based on these reactions. Not only does it infer those pathways, but it determines the abundances of those pathways in each sample. So then not only can we have the enzyme commission numbers or the predicted enzymes present in each sample, but we can also have the predicted metabolic pathways per sample. And so what this is going to do is it's going to predict the ASV contributions to each pathway abundance for the entire community. Okay, so it's going to predict ASV contributions across all ASVs for every sample. And it's possible to predict pathway abundances per ASV rather than like per sample, but it's really computationally intensive, and it was not one of our goals for DOG, which is the group that I'm working with. But just so you know, it's possible to have that analysis as well. So the previous table that I showed you and walked you through was the one at the top, where we've got the predicted EC relative abundances per ASV in each of the samples. And so that table is on top, and then we now have this predicted metacyc pathway abundances table at the bottom. And so the calculations are the exact same, where we've got the taxon abundances, normalized taxon abundances of each ASV in the samples, and we've got the predicted functional count for the genome. So this would be the metacyc pathways. So for each genome, it's expected that there's 0.65 of this pathway IC-MET2 per genome. So then we would take 19.33 and multiply 0.65, and then we would end up with an expected number 12.6 of this IC-MET2 pathway, and sample 114S1 is coming from ASV 1037. So just some final steps here before you can start, you can start processing, analyzing the data, is to add some descriptions to the two tables that you've generated. And this is done really easily by just adding descriptions.py. And you'll see why this is important just for your records to have this, because it's a really easy way to go back and check, okay, well, yeah, I have the CC number, but what enzyme was it classified as? So these resulting output files would look like this, where we've got predicted pathways on the left. So this is basically an OTU table with an extra column for description. All right, so we've got IC-MET2 pathway, and that is supposed to be this N10 formal tetrahydrofolate biosynthesis pathway. Okay, so it's just an extra added column. It's really easy to refer back to whenever you're starting to look at the results of your analyses. And similarly for this EC table on the right, we've got this extra added column of description where we've got like EC 1.1.1, which is supposed to be an alcohol dehydrogenase. So you can easily bring these files, these EC and metaseq pathway abundance tables into R and make a phyloseq object because, well, it's just a lot easier to work with phyloseq object than it is with just OTU tables, in my opinion. I would caution you to look through the literature to make sure that the specific statistical analysis that you're running on these tables is appropriate. There is a lot in the literature to help guide you to make the best decision. And again, make sure that whatever you're doing, your analyses include some sort of a normalization step, whether that be transforming into relative abundances or z-scores or centered log ratios. All right, good luck.